I'm Monsuru Adebowale. I'm a third-year PhD student at the University of Bristol. And the topic of my presentation is Trouble in the Ocean, a PETM record captured by planktic foraminifera species. Climate change has been changing the ecosystem, and it's changed the physical and chemical processes in the ocean. And then we also have natural processes like carbon and hydrological cycle being affected. And if you look at the present and then the future image that we have, you can see that carbon dioxide will increase in the future, leading to acidification of the ocean. And then this acidification of the ocean will lead to carbonate dissolution in the ocean. And then during this high temperature or then warming, we'll have the wind and then wave action will be affected, which will lead to stratification of the ocean. And then stratification of the ocean will lead to anoxia, especially in the deep ocean. And then also nutrient supply from the deep sea to the surface will also be reduced due to reduction in upwelling, and then on other, also affecting other processes in the ocean. And then the climate change is also impacting our ecosystem biologically. Like you've seen in the image on the left, on the right-hand side, you can see bleaching of coral, and then this has been abandoned by marine organisms. And then behind that, we can see the picture of a healthy coralline algae, which is serving as a good habitat for marine organisms. And also, calcification is also reduced during this climate change impact. And then we have an image on the left-hand side from Fox et al. 2020, which shows that HM Challenger expedition in the late 19th century, and then Tara expedition in the early 21st century. And we can see that there's decrease, in about 50% decrease in foraminifera shell thickness, which shows that its impact is changing the ecosystem biologically. And then we can also have migration of species when they are stressed, and this will also affect carbonate distribution in the ocean. So complementing field observation and lab-based experiment, which are not probably the best way of looking at the response of marine organisms, because laboratory experiments are short, and then that will also be limited in understanding the evolution or ontogeny of this species, and then the potential for adaptation will also be minimized. And then there will be reduction in assessing regional variability at different locations around the globe. And then looking at being able to access multiple range of environmental driver will also be limited. And then we can look at the geological record of climate change, which can serve an analog to future anthropogenic change. And the aim of the study is to test the impact of natural rapid environmental change on marine plankton system. And then we look at foraminifera carbonate production, the size distribution of forams to assess stress, and then foraminifera weight in order to quantify calcification, and then their roles in the biogeochemical circle. So we will look at the hypothermal events in the Eocene, and the image on the left-hand side shows the Paleocene-Eocene thermal maximum, which is showing the negative carbonate excursion during the PETM, as we can see from the image B and C, looking at the oxygen and then the carbonate isotope. And then this is reducing after the CIA to the recovery and then probably to the background level. And column A shows the typical PETM core, which is showing the contrast between the carbonate layer before the CIE and then the clay layer at the beginning of the PETM. You can see that on the red dashed line that we see, and then the PETM is about 56 million years ago. And then the image at the middle is showing carbonate isotope of these two hyperthermal events in the Eocene, showing the magnitude of the PETM and then that of the ETM. And then we can see that the ETM precedes the ETM2 by about 2 million years. And also the magnitude of the PETM is twice that of the ETM. And then also the duration of the ETM is half that of the PETM. And then benthic foraminifera extinction were recorded during the PETM and then none during the ETM. So the methodology we employed, we looked at sand percentage of foraminifera and also their carbonate accumulation. And then we look at fragmentation in order to assess a carbonate dissolution of a foram, and then size analysis, which are carried out by automated microscopes, which counts thousands of foraminifera specimens. And then after that, we process this foraminifera specimen in order to remove any fragment, benthic organism, diatoms, obstacles, just leaving mainly planktic foraminifera. And then afterward, we process this, leaving about 800 to 3,000 specimens for a size measurement. And then size analysis, which I've said earlier, we want to quantify classification. We looked at open layer species, the most abundant of these species. But open layer species, we looked at Monsuru sabatina, and then the thermocline species, we looked at Sabatina patagonica, and then we measure the weight of these two species. Hypothesis, we hypothesize that if sizes are larger in the tropics, and then this species migrates away from this environment due to stress, and then moving into a favorable environment, this leads to size increase in the poles, and then stress at the tropics will also lead to dwarfism, and thermal and ocean acidification will also reduce carbonate production. So the PETM result, the first row will have it's at site 1135, which is at the Kwajalein Plateau in the Southern Ocean. And then we can see in this second column that fragmentation is a little stable, just slight increase during the PETM, the red or pink line, and then the green line is the recovery interval. And then you can see that size percentage is also fairly stable, only a reduction at about 1,050 years before the CIE, and then the mean size is also stable, like we have seen, and then averaging about 320 or so at this side, and then the middle row shows site 1209 at Shansky Rise, the Equatorial Pacific site, which also shows that fragmentation percentage is also stable. But if you look at the sand percentage, we can see that sand percentage increases during the CIE than before and then beyond the CIE. And then the size, mean size here at this site, it's more variable, and like we can see, and then the average size at this size is about 370. And then going back to the last row where we have site 1409, which is the Newfoundland site in the North Pacific, we can see fragmentation peaks at about 80%, which we can say upscale, and which we can say we can relate that to the carbonate isotope at the PETM, and then also there is a size sound percentage reduction during this interval. And there's also fairly variability of mean size, like you've seen in the diagram, and then it averages about 350 micrometer at this site. So looking at the foraminifera weight, we can see that the weight is fairly stable at the shallowest site 1135, just some changes before the CIA. But looking at the other two sites, site 1209 and then site 1409, we can see that the foraminifera weight increases during the PETM, and this is not expected. This is strange. So we have to carefully break the samples and then review them under using the scanning electron microscope, and then we see that these are filled with other material which compromises the weight of the material. So these cannot be trusted. And then going to the ETM result, we can also see from the Equatorial Pacific side that the fragmentation is also fairly stable, and then that at the core ETM2, we can see size reduction from the before the core ETM2, and then also the availability in the main size, averaging about 370 around this site at this event, and then site 1409 also shows that there is sensibility of environmental change at this site, because you can see some slight increase in fragmentation at this site, and also sound percentage reduction. And the average main size at this site is about 350. So we try to upscale our results to global level, and then we employ the forum ecogeny model in order to look at what will happen, what we expect globally. And then this is a trait-based model which depends on size. Size is the master trait. And then, like you can see in the left-hand side, the diagram shows smaller plankton will grow faster, because at smaller sizes, we have bigger surface area. And then the bigger ones will hit up the smaller one, like we've seen in the diagram below. The big fish eating up the smaller ones. And then we employed foraminifera, non-spinous foraminifera is part of the zooplankton in the plankton community model. And then foraminifera size and biomass, if you want to look at the response of size and the biomass during the PETM. And then we put this model into test, and then we can see that we compare this to modern work that has been published by B and then Toddle in 1971, and we see that there's increase in the biomass distribution in the Southern Ocean, just like the model also tells us, and then also around the Arctic Ocean, and then somewhere in the Indian Ocean around the east coast of Africa. So this shows that foraminifera biomass distribution is well predicted by this model. And then the impact of climate change during the PETM, the model shows that there are no changes in the biomass around the study size, and then also there's no significant mean size and change across the sites. This is because the model does not employ symbol-bearing foraminifera species, so it shows no difference in this location when you look at the mean size. But the model shows increase in biomass and then mean size around the 30s, like we've seen in the color, and the red color around the 30s, which shows that there is increase in biomass distribution and then mean size. So in conclusion, carbonate dissolution is more severe during the PETM in the Atlantic Ocean probably due to the proximity to volcanism, and then size increases from pole to the tropics, and there are no clear indication of size increase across all sides, and then during the interval. This is supported by the model. And then larger global mean size is witnessed around the 30s during the PETM. And then there is increase in foraminifera contribution to the carbonate in the tropical sites. Overall, foraminifera resilience is about 10 times lower than what we witness in climate change today. So thank you for the opportunity, and thank you everyone for listening. So the question.
Thank you so much, Monsuru. We're actually going to have to move on. There's a question for you in the chat there, but with the interest of time, we'll have to line up our next speaker. But thank you so much.
All right.
So I believe next we have Erik Sperling. I'll just get your slides up for you. Super, they should be visible now. Go ahead.
Great. Well, it is awesome to be here at Palace without the long plane flight, although, of course, I miss seeing all of you in person. Today, I'm going to be talking about the Metabolic Index, and I'd like to thank a number of people at both Stanford and University of Washington who have been involved with this, as well as our funding sources. So this photo is from my master's research, where I looked at the Permian-Triassic mass extinction in the Quinn River Formation in Nevada. And in this locality, this section goes from well-oxygenated late Permian cherts into dark anoxic early Triassic shales. And this is part of a worldwide anoxic event that's been recognized by people like Paul Wignall and Richard Twitchett. Since then, we've had a number of sophisticated geochemical studies that have both confirmed that the Permian-Triassic extinction is associated with warming and anoxia, and also given a much better look at the magnitude of the change, such as shown by these figures on the left. And these come from a paper that I'll be talking a lot about today, the Penn et al. 2018 paper in Science. And what they're showing in the top figure is the magnitude of temperature change across the Permian-Triassic mass extinction from Conodont delta O18 values. And on the bottom panel, the percentage of seafloor anoxia from carbonate uranium isotopes. So these data show that the Permian-Triassic extinction is associated or correlated with these, but there's still a number of questions as paleontologists that we'd really like to know. So for instance, were both of these kill mechanisms? Was one of these worse than the other? And was there spatial variability in the biological impact of these different stressors? And I think the crux of comparing geochemical data with the paleontological record and thinking about how environment affects biology on Earth is well encapsulated in this quote in this 2013 paper by Andy Knoll. So he writes, "We can use the fossil record illuminated by comparative biology to reconstruct a narrative history of life. And increasingly, geochemical analyses of sedimentary rocks are revealing a history of both long-term environmental change and transient perturbations to the Earth's system. Biological and environmental history can, in turn, be linked by stratigraphic correlation. But to interpret correlation in terms of causal mechanisms, we need a bridging concept. Here, I argue that physiology provides the necessary bridge." So in other words, he's saying that most of the time, what we're just doing is we're looking at wiggles in the geochemistry, we're comparing it to the fossil record, and we're drawing correlations between them. But that doesn't get us towards causality. And so today I'm excited to talk about the Metabolic Index, which is an ecophysiological framework that I think can help us bridge those geochemical and paleontological records. And because a lot of the papers on the Metabolic Index are relatively dense, today I'm going to try to walk you through a little bit better what the Metabolic Index is, as well as talk about some of the warts and some of the future directions we're trying to take it. So the Metabolic Index, as first put forward in this Deutsch et al. 2015 paper, is basically just a ratio of oxygen supply to the organism versus its resting demand. So you can ignore all the math that I'm going to show you. That's basically just what it is: the ratio of oxygen supply to resting oxygen demand. So in this paper, Deutsch et al. Established supply terms that are related to body mass and oxygen availability, and demand terms which are related to body mass and temperature. And they put these together in the Metabolic Index, which is encapsulated in this term of phi, which, as I said, is simply the ratio of oxygen supply to resting oxygen demand. In order to calculate phi for a given organism, we need to know four things. The first is the hypoxia tolerance, or the oxygen level at which an organism can no longer maintain its standard metabolic rate, which we refer to as A0. We need to know the temperature sensitivity of this oxygen demand, which we refer to E0. And these two values are obtained through respirometry experiments. And shown here on the top left are my students Tom Boag and Andy Marquez conducting respirometry experiments on brachiopods at the Friday Harbor Marine Labs. And basically what you do here is you put an organism in a respirometry cell and measure the oxygen drawdown through time using optical oxygen sensors. And that's shown in the center top panel. So you see the oxygen drawdown proceeding at a steady rate. This is just its standard metabolic rate. At some point, though, the level of oxygen that's left isn't enough for the organism to meet all its energy demands using aerobic metabolisms, and so it switches over to anaerobic metabolisms. And the rate of oxygen consumption with time drops dramatically. And we can label this the critical oxygen level, or PO2 crit. We then repeat these experiments at a bunch of different temperatures at a bunch of time. This is shown on the right for a sea anemone and Matostella vectensis. And we get that temperature sensitivity of oxygen demand. The next two things we need are the environmental oxygen levels and the environmental temperature. And we can get these either through measuring in the modern ocean or from Earth system models if we're interested in pushing this forward or back in time. The final thing we need to know, I told you that phi represents the relationship between oxygen supply and resting demand. So phi level of 1, an organism is going to be able to perfectly satisfy its resting demand, but it's not going to be able to do things like feed or defend itself or reproduce or etc. It needs some multiplier on that, which we refer to as phi crit. And this is measured through looking at the biogeographic limits of the organism. So I'm going to illustrate this a little bit more using the work of my postdoc, Murray Duncan, who during his PhD worked on this fish, the Roman sea bream from South Africa. And South Africa has a really interesting oceanography. Shown here on the top left is temperature, very cold levels around the southern tip of South Africa, with temperature increasing along both of the coasts, but much faster along the east coast as you go into the Indian Ocean. Oxygen is quite low around the southwest coast and up the west coast, and this is related to the Benguela current oxygen minimum zone. So Murray measured the physiology of these organisms, he plugged them into the metabolic index equation, and he was able to calculate phi values for the sea bream along this entire area. And you see that the highest values are contained down around the south coast, with phi levels decreasing up the west coast due to lower oxygen, and up the east coast due to warmer temperatures. Murray was then able to look at the biogeographic limits of these species and calculate a phi crit level, or a multiplier on that minimum oxygen requirement of about 2.8. He was then able to create a map of suitable aerobic habitat in the modern ocean, which is the top right panel. And then he was able to take a projection for the year 2100, an oceanographic model, and predict where suitable aerobic habitat for this species is going to persist. And it's a little hard to show without a laser pointer, but the edges of the species range are predicted to contract. And in particular, the suitable habitat between Port Alfred and Port St. John's is predicted to be wiped out. So this is how neontologists are using the metabolic index. Of course, we wanted to project this back and use this to examine events in the fossil record. And particularly, the first one we applied this to in this Penn et al. Paper was the Permian-Triassic mass extinction. So the first part of this study, what the UW team did, was they made a model of the in-Permian ocean using the community Earth system model. And some of those results are shown on the left. You don't need to really understand any of the details here. The point I want to make is basically that ocean modelers are getting increasingly good at modeling ancient oceans. And this is really important, simply because what controls aerobic habitat viability is really the synergistic combination of oxygen and temperature, as shown with that example with the Roman sea bream. Modern oceans are heterogeneous with respect to oxygen and temperature. Ancient oceans are heterogeneous with respect to oxygen and temperature. And taking into account these heterogeneities, both in terms of the spatial distribution and the bathymetric distribution of oxygen and temperature, is becoming really important. So after creating these models, we then populated the in-Permian ocean with theoretical ecophysiotypes that had physiological traits drawn from the distribution in the modern ocean. We then perturbed this ocean to a state matching that early Triassic geochemical data that I showed earlier. And this was done with a huge CO2 spike meant to stimulate the Siberian traps. And then finally, we calculated the ecophysiotype extinction versus latitude and compared it to new analyses of the fossil record. And these results are shown here. So what's shown in the black line is our model ecophysiotype extinction, with the gray bars representing different sensitivity analyses for how much of the aerobic habitat volume of each species must disappear in order for that ecophysiotype to go extinct. And this is compared to the fossil record, which are these red dots. And you see that the model ecophysiotype extinction does a good job of reproducing the fossil record, and in particular, the biogeographic selectivity of the extinction, with higher rates of extinction in the polar regions as compared to the tropics. And so at a first level, this suggests that the synergistic combination of oxygen and temperature change, or what we call temperature-dependent hypoxia, is likely the main driver of the Permian-Triassic mass extinction, as opposed to something else like ocean acidification. But the metabolic index can also give a lot of insight into other things, such as what is the cause of that biogeographic extinction, biogeographic selectivity that wouldn't necessarily be apparent from the fossil record. And what our analysis suggested is this extinction pattern is actually driven by the Permian distribution of ecophysiotypes in the pre-extinction ocean. So what this graph is showing is the average hypoxic threshold and the average active to resting rate, or phi-crit, of ecophysiotypes in the in-Permian ocean. And you can see that the polar regions have much higher values of these, suggesting that polar organisms have much higher oxygen demands than organisms living in the tropics. Or in other words, organisms living in the tropics are kind of pre-adapted to warm, low oxygen conditions. And when these conditions arise globally, they're either able to deal with these conditions or to move to track these habitats. Whereas extinction or organisms in the poles have simply nowhere else to go. We're also able to get insights into the specific contribution of oxygen loss and warming by looking at things like regional extirpation. And this particularly suggests that the extirpation was caused by oxygen loss in the poles and warming in the tropics. There's a bunch of sensitivity analyses in these papers. In this paper, basically each one of these analyses is an entire paper in and of itself, and I really encourage you to check it out if you're interested. All right, so obviously I'm a big cheerleader for the metabolic index, but I did want to talk a little bit more about some of the issues we're currently facing, and how we're trying to address this. So the first thing is with respect to cooling. These are some data from a paper by Tom Boag looking at oxygen, temperature, and the early evolution of the Ediacara biota. And what they're showing for this anemone is the critical oxygen level shown in blue against temperature. And you see it increasing with temperature, which is what you'd expect based on the fact that metabolic rate is also going up, shown by these orange dots. The problem is, if we look at the cool side here, what we see is in fact a V in terms of these critical oxygen levels. And this is occurring in spite of the fact that the metabolic rate is continually going up over this temperature range. So why is this happening? Well, the warm side is easy to understand. It's simply higher temperatures, increasing biochemical reaction rates, raising the standard metabolic rate, and driving a higher PO2 crit. On the cold side, what we believe going on is that actually the oxygen supply to the organism is decreasing in cooler waters due to the effect of temperature on oxygen diffusion rates. Also, the organism is having to pump really hard to move this cold, viscous water along its respiratory surfaces. And so we actually see a higher increase of those PO2 crits at colder temperatures. We're working on modeling this and incorporating this into the metabolic index. But the take-home message for now is that the metabolic index works really well for warming events, or we think it does, and not so well for cooling events. Next, we've only been incorporating the metabolic index in a time-dependent fashion. So we've been comparing kind of a before and after state. We haven't been really incorporating anything about dispersion or adaptation rates, or really the macroecological principles that you'd like to incorporate. Although I would note, if we're doing the kind of standard wiggle matching between geochemistry and the fossil record, those effects are also not explicitly incorporated. In any case, this is something that Rich Stockey is hoping to address during his postdoc, and if you're further interested in this, I would encourage you to talk to him. The final thing is that almost all of the data that has been generated by neontologists for physiology is from economically important fish and decapod crustaceans, and we actually don't have a great sampling of what's makes up the bulk of the fossil record. And so my lab has been spending a ton of time at marine stations like Friday Harbor, trying to fill in these data, and I think we're going to find some really interesting things. And I'd specifically like to thank the Paleontological Association and NSF for support for this. Ultimately, though, we expect these data are going to give us a better resolution of the events rather than being any sort of game changer. And I just show here 4 supplementary analyses from the Penn paper. Each of these are drawn from a different quadrant of the E0 versus A0 physiological space. And you see that each of them are reproducing the same basic pattern of the end-Permian extinction, even if some of the details are changing between these analyses. So in conclusion, I think the metabolic index shows a lot of promise in terms of connecting environmental change with biotic response. And I hope you'll agree with this, and I look forward to seeing what Rich Stockey has to tell us about early Paleozoic extinctions. Thank you very much.
Super, thank you so much, Erik. Probably in the interest of time, we'll probably move on without questions. I think you're absolutely right. There's obviously these limitations to the physiological measurements from modern species, particularly I think in deep waters as well. So we'll move on to Richard Stockey is actually going to tell us a little bit about that work and talk a little bit about oxygenation in the Phanerozoic. So if I start off your slides. There you go.
Thank you. Okay. So today I'd like to talk, sort of building on from what Erik's just been telling us all about, talk about how we might use these principles of ecophysiology combined with system modeling to try and investigate the links between Earth surface oxygenation and metazoan ecophysiology and their impacts on extinction intensity through the Phanerozoic. So here I'm showing a plot of extinction rates through the Phanerozoic. The line and the points represent the mean of 12 different methodological approaches employed using the DivDynR package, and the gray envelope represents the maximum and minima of each of these values at the stage level. Here we're using the PBDB for basically to compute these extinction rates using all marine animal occurrences through the Phanerozoic. And what I'd really like to draw your attention to here is the relative frequency of high magnitude extinction rates, and particularly the relative frequency of extinctions outside of the canonical Big Five. Notably, we see similar trends if we use the Sepkoski database, but really these trends of decreasing extinction rates through time seem to be a robust feature of the marine fossil record, and particularly these really high Cambrian Ordovician extinction rates, which I want to talk more about today. And what I want to look at further is the underexplored correlation between high extinction rates in the early Paleozoic and limited atmospheric oxygenation in the early Paleozoic. So traditionally, we view atmospheric oxygen, or we have viewed atmospheric oxygen as having risen in 2 steps. One at the beginning of the Proterozoic, the GOE, and another Neoproterozoic oxygenation event towards the end of the Proterozoic. However, there's increasing agreement between biogeochemical modeling and the geochemical proxy record that actually in the early Paleozoic, atmospheric oxygen levels were probably below 50% of modern. And we didn't hit modern levels of atmospheric oxygenation until sometime between the Ordovician and the Devonian. So we have this correlation between limited surface oxygenation and high extinction rates in the early Paleozoic. But as Erik was just talking about, what we're really interested in doing is trying to move beyond simple wiggle matching. So I want to talk to you about some work we've been doing to try and get further. So if we're interested in looking at the impact of environmental change on marine biodiversity dynamics, in the modern, we're overwhelmed by data and we're overwhelmed by the pressing issue of time. But doing this kind of science is relatively straightforward. We can perform long-range monitoring studies and we can perform laboratory experiments looking at the impacts of environmental variables on the physiology of marine animals. But as Earth historians, this really boils down to an issue of separating causation from correlation. And to try and move beyond this wiggle matching, we've been developing a workflow to try and understand how we can separate causation from correlation. So if we're interested If we see paleobiological change and geochemical change in the geologic record, what can we do to unravel whether or not those two things, the geochemical change we see, might have been driving the paleobiological change we see? That might begin by generating new geochemical and paleobiological records. Beyond that, what we've been trying to do is generate a mechanistic framework for understanding how ancient animals living in ancient oceans would actually have interacted with their environments. So from a geochemical perspective, this involves developing statistically robust models of what geochemical proxies actually mean for ancient marine environments, and specifically trying to calibrate the uncertainty on what those proxies mean for ancient marine environments. From the paleobio side, what it means is trying to understand how animals might actually have responded. And as Erik's mentioned, we've been borrowing a lot from techniques in modern ecophysiology. And then our system models are really the glue that holds our whole approach together. Firstly, our system modeling allows us to generate three-dimensional ocean models at reasonably high spatial resolution that can translate our geochemical proxy records into a realistic array of feasible ancient ocean environments. Secondly, that high spatial resolution allows us to write ecological models that allow our simulated organisms to interact directly with those marine environments. So Erik mentioned the metabolic index. The metabolic index is built on this principle of temperature-dependent hypoxia, and it's been well documented in modern marine ectotherms. We're working on documenting it in even more in our lab, and it really describes the synergistic relationship between seawater oxygen and temperature on the minimum requirements for animal metabolism. And specifically what we see is that the minimum required partial pressure of oxygen, or minimum required PO2, increases exponentially with temperature to support resting metabolism. To support active metabolism, or more specifically, ecologically sustainable populations, we require an increase in that PO2, which results in steepening of that exponential relationship. And these exponential relationships make a fundamental prediction about the thermal safety margins of marine ectotherms living in ancient oceans. So here I'm comparing a cold adapted species living in the modern to one living, say, at 40% present atmospheric oxygen levels, so sort of a Paleozoic style atmosphere. And as you can see, if you're an ectotherm living under much lower levels of atmospheric oxygen, you're likely to be much more vulnerable to a global warming event. So in our models, or in the models that I'm going to present here, we're using a probabilistic representation of the metabolic index. So we have probability distributions describing three ecophysiologically important variables defined in the metabolic index. These are based upon experimental respirometry and upon the biogeography of those taxa. And then for each of our analyses, we're generating populations of 1,000 synthetic ecophysiotypes. So the CGenia system model provides a brilliant arena for our simulated ecophysiotypes to interact directly with ancient modeled marine environments. CGenie was developed by Andy Ridgewell as a spatially explicit model for the biogeochemical characterization of paleooceanographic problems. And what this allows us to do is take simulated oxygen and temperature profiles for hypothesized ancient oceans and calculate the percentage of our ecophysiotype distributions that would have been viable in each cell of our ancient ocean. So here I'm just plotting that as a heat map of percentages for the surface ocean with an Ordovician tectonic configuration as an example. One of the awesome things about CGenie is that we can use lots of different tectonic configurations, try and understand the impact of that on our simulations. In the analyses I'm presenting today, I'm going to talk about shelf environments only, because that's where we find the majority of marine biodiversity today, and it also constitutes the majority of our fossil record. But please note that we see very similar results when we look at the entire ocean model. So let's return to our question. Can limited atmospheric oxygenation explain high early Phanerozoic extinction rates? To do that, we've used an ensemble Earth system modeling and ecophysiological modeling approach. So for each of 5 different ensembles. We've forced C. genie at a realistic range of atmospheric oxygen concentrations and atmospheric CO2 concentrations for the Phanerozoic. And then we've applied our ecophysiological model on the final time step of our steady state ocean, which takes 10,000 to 20,000 years to reach steady state, depending on the configuration. And as I said, we performed 5 different ensembles. So 3 of these look directly at different continental configurations. We've used an Ordovician configuration, a Permian configuration, and a Paleocene configuration to try and get an even spread through the Phanerozoic. And I've also performed sensitivity analyses looking at solar luminosity, which is going to affect our latitudinal temperature gradients, and also calm remineralization depth, which is going to impact the depth profile of oxygen in the ocean. And critically, we expect all 3 of these variables, continental configuration, solar luminosity, and calm remineralization, to have changed through the Phanerozoic, or they've certainly been hypothesized to. So testing these is critical to isolating the impacts of atmospheric oxygen specifically on our extinction events. So now onto some results. Here on the x-axis, we have atmospheric oxygen in present atmospheric level units. And then on the y-axis, we have what we're calling metabolic habitat viability. So this is just the percentage of our simulated ecophysiotypes that can live anywhere in the shelf environments of our genie oceans. And here, each of our individual points represents an individual ocean model that's been run for at least 10,000 years. And I've color-coded them based on mean equatorial surface temperature, which is forced by carbon dioxide concentrations. And what I really want to draw your eye to here is the difference in proportional loss of metabolic habitat viability between colder and warmer states at modern levels of atmospheric oxygen versus reconstructed early Paleozoic levels of atmospheric oxygen. As you'll see, there's much greater spread at early Paleozoic levels, and also because we're starting from less metabolic habitat viability to begin with, the proportional loss of metabolic habitat viability is even greater. So this supports this idea that animals are living much closer to their thermal safety margin at lower levels of atmospheric oxygen, specifically the kind of levels that we've reconstructed for the early Paleozoic. Just looking briefly at our sensitivity analyses, looking at the Permian, Paleocene, and then shallow remineralization depths and modern solar luminosity in the Ordovician, we see very similar patterns. I don't have time to go through all of these individually, but I think you'll be able to see for yourself that there's a much greater spread in all of these at early Paleozoic levels of oxygen versus modern. So then we're specifically interested in extinction here. So I've been modeling individual warming-driven extinction events of approximately 5 degrees warming from a modern baseline. Ideally, I'd like to represent something along the lines of the warming that we see across the Paleocene Eocene thermal maximum, at least broadly. So again, we have atmospheric oxygen on the x-axis, and then we have global ecophysiotype extinction on the y. So simply the proportion of ecophysiotypes that are viable in our colder model that then go extinct in our warmer model. And I've color-coded this based on the 3 continental configurations that we use. As you'll be able to see, we see increasing global ecophysiotype extinction with decreasing atmospheric oxygen. And notably, we see an inflection in this increasing ecophysiotype extinction as we approach roughly early Paleozoic levels of atmospheric oxygen. So again, the early Paleozoic was a really bad time to be trying to survive a warming event. So here we're presenting a clear link between atmospheric oxygen levels and vulnerability to warming-driven extinction. And we therefore put forward that atmospheric oxygen was a fundamental modulator of extinction vulnerability through the Phanerozoic and was likely a primary driver of these high extinctions that we observe in the early Paleozoic. And more broadly, we just want to highlight the metabolic index as an awesome way to go about trying to relate these geochemical records to the biodiversity dynamics that we observe in the fossil record. So just to conclude, atmospheric oxygen is a first order control on extinction vulnerability of marine animals under standard climate variability. And as I said, those high early Paleozoic extinction rates can be mechanistically linked to limited atmospheric oxygenation. And then the metabolic index is just an awesome tool that we can begin to use to bridge the links between the fossil record and the geochemical record. And once again, we're highlighting the real importance of the synergistic effects of ocean deoxygenation and ocean warming on marine animals, as we're observing in the modern and as we're increasingly seeing, was probably a huge problem at different times in the geologic past. With that, I'd like to thank all my funding sources and these people from Stanford and University of Washington, and I'll happily take any questions if we have time. Thank you.
Wonderful, thank you very much, Richard. We actually have got one question in the chat here, just quickly, from Daniela Schmidt. So Daniela says, Gini doesn't actually perform very well in coastal settings. So how much do you think that might impact your model comparisons?
Right, that's a great point. Thank you. So Gini is often kind of, people criticize Gini for having issues in shallow marine settings. That's part of the reason that we've been working with multiple continental configurations to try and see what different dynamics of shallow seas, where we had loads of shallow seas in the Ordovician, and perhaps and the Permian, and perhaps had less in the modern. And it's also why we've been repeating all these analyses through the global ocean. So we have sensitivity analyses just looking at the surface ocean, and we see very similar trends. So the resolution of Gini along the coast doesn't seem to be sort of really changing our ecophysiological analysis.
Super. Wow, thank you very much.